Very good afternoon to everyone, and thank you for joining us fortnightly session organized by Knowledge and Awareness Mapping Platform. I, Arika Mathur, convene moderate this session today. So, as our special guest, Mr. Gautam Dongre, NASCO Board Member, GAS CD, Tanika Mendiratta, Project Associate, Patela, PhD Scholar, Ms. Megha, PhD Scholar from Dr. Debojyoti Chakrabarti, CSIR IGIB New Delhi. Mr. Ashish, Project Director of CAMP, Mr. Amit Shuk, Capacity Building Group, and my fellow champions. I bid you a mapping platform is an initiative and not NISPR, that is National Institute of Education and Policy Research Technology with industrial partner NCPL. CAMP believes is blessed with a core talent and aims to them through this platform. CAMP has organized its first scientific excursion at CSIR, Institute of Genomics and Integrative Biology, IGIB, New Delhi, on 8th of September 2022, where more than 150 participants, including students, parents, and teachers, witnessed the zebrafish facility, transmission electron microscopy, and scanning electron microscope. CAMP is back with the 12th fortnightly knowledge sharing session for the year 2022 on sickle cell disease. In this session, we will share with you what is sickle cell disease and why is it important to know about it. So sickle cell disease is an inherited blood disorder that affects red blood cells. People with sickle cell disease have red blood cells that contain mostly hemoglobin S, an abnormal type of hemoglobin. September is National Sickle Cell Awareness Month, and as we continue to fight with COVID-19, SCDAA is compiling resources to assist the sickle cell community during the pandemic. So we need to increase the awareness about the sickle cell disease. So before we go ahead and invite our presenters here and our special guest, please do not forget to give your feedback about this workshop at the end of this session so that we can keep improving our content. The feedback form can be shared in the form of a video or in the form of a Google form that will be shared in the chat box at the end of the session. Stay tuned till the end of the session to participate in the fortnightly quick challenge where you can answer a simple question and be the lucky winner. The quick challenge link will be shared in the chat box at the end of the session. The winner will be selected randomly based on the criteria defined in the participation form. Stay tuned and stay with us till the end of the session. Also, please share your question and answers about the workshop in the chat box, or you can raise your hand after the session, which will be taken up during and after the session by our presenters. So we hope that you will enjoy this session. Without the further ado, I now invite Ms. Kanika Mendiratta, Project Associate of Debjyoti Chakrabarti Lab, CSIR, Institute of Genomics and Integrative Biology, IGIB, New Delhi. CSIR, Institute of Genomics and Integrative Biology, is a premier institute of Council of Scientific and Industrial Research engaged in research of national importance in the areas of genomics, molecular medicine, bioinformatics, and pre-genomics. The mission is to translate concepts developed in basic biological research to commercially viable technologies for healthcare. So now I request Ms. Kanika to kindly address the gathering.
Hello, everyone. I would like to share the screen first. We have a PPT made for you guys. I hope everybody can see my screen. As has been introduced already, thank you so much. We are working in the sickle cell disease domain under Dr. Debojyoti Chakrabarti's lab at CSIR IGIB. And sickle cell disease is actually the most common monogenic disorders in the world. and has a very high prevalence in Indian population, especially in Indian tribal population. So today we'll be talking about how we started working in this area of research, what is the pathophysiology, the symptoms, and current treatment strategies that exist for the condition. And we'll also move on then to the therapy that has been developed by scientists at our lab, which is gonna go into a clinical trial phase very soon. We'll also talk about the Feluda kit that our lab had developed during the pandemic period and how we utilized it to test for samples for SARS-CoV-2 virus. So since it has already been told, it's the Sickle Cell Disease Awareness Month. And as part of this month, we are we've organized this webinar with CAMP. I'd like to start by introducing Gautam sir. Gautam Dongre is actually a member of the NASCO, and he has been working in the sickle cell disease area for quite some time. We would like to take up your questions at the end of this presentation, and the topic might get a bit more scientific as we move forward, but we are open to questions even after the webinar is over. So Gautam sir, if you are there, if you can hear me, I would like to ask you some questions about the work that you are doing. Gautam, are you there?
Yeah.
Yes, sir.
Am I audible?
Hi, sir. So I wanted to ask you, how did your journey began in the field of SCD?
Yeah.
आपके बारे में कुछ?
जी 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 हिंदी में कंफर्टेबल रहेगा आप सभी को?
Yes, sir, you can do it, and I'll help later with just a summary of whatever you say.
Okay, I will try to speak in English also. So it might be some difficult to understand to you my English, but I will try. Okay, thank you so much. So my journey start. Yeah, please.
I'm so sorry. We can have a mixed language. That is okay, sir. We have students joined us from different parts of India, so we can have English and Hindi both. We can have a mixture.
Okay, I am more comfortable in Hindi, so that's why I requested you.
That is also absolutely fine, sir. We'll do it.
Okay, thank you so much. तो सिकल सेल डिसीज के साथ जो मेरी जर्नी है वो आज से 18 साल पहले शुरू हुई, जब मेरे घर एक सिकल सेल पीड़ित बच्चे ने जन्म लिया, यानी मेरा पहला बेटा गिरीश। इस पिक्चर में जो आप तस्वीर देख रहे हैं पहली, वही मेरा बेटा है जिसका नाम है गिरीश, और वो एक पेन क्राइसिस के समय हॉस्पिटल में एडमिट था, और वो दिन था राखी का समय, राखी का त्योहार। अभी इसी जो राखी में गए थे उस दिन की ये तस्वीर है जो मैडम ने यहाँ पर यूज किया। और वो जो स्टाफ नर्स के ड्रेस में है वो मेरी वाइफ है, यानी गिरीश की मम्मी, जो कि गवर्नमेंट मेडिकल हॉस्पिटल में स्टाफ नर्स है। और जो राखी बांध रही है वो मेरी दोनों भतीजियां हैं, जो कि गिरीश की कजिन सिस्टर्स है। तो हॉस्पिटल में आकर के उन्होंने राखी के दिन उन्हें राखी बांधी। तो मेरी जर्नी 18 साल पहले शुरू हुई, जब मुझे गिरीश जन्म में आया, और होने के बाद से ही उसे तकलीफें शुरू हो गई। सिकल सेल डिसीज क्या है इस पर तो आगे आपको बताया जाएगा, लेकिन इस डिसीज में बहुत ज़्यादा पेन होता है बॉडी में, और ये जेनेटिक है, जैसे कि मैडम ने कहा है, हेरिडिटरी है। ये आपको ये जो बच्चे को होता है ये मम्मी और पापा इनकी वजह से ही होता है, और मदर के पेट में ही होता है, और दूसरा कोई इसका रीजन नहीं है। तो मेरे बच्चे को भी मेरी और मेरी वाइफ की वजह से सिकल सेल डिसीज हो गया, क्योंकि ये होने का कारण होता है कि हम दोनों सिकल सेल ट्रेट है, यानी हमारे दोनों के जीन्स में सिकल सेल के थोड़े थोड़े अंश थे, जो कि हमारा बेटा होने के समय में वो दोनों अंश मिल करके उसको ज्यादा हो गया, जिसको डिसीज कहा जाता है। तो बच्चों, कॉमन लैंग्वेज में हम हमारे माँ बाप से बहुत कुछ लेते हैं जब इस जिंदगी में आते हैं। जैसे आप देखोगे कि आपका चेहरा या तो पापा से मिलता है या तो मम्मी से मिलता है ना, या फिर आपका कलर पापा से मिलता है, मम्मी से मिलती है, आपकी हाइट मम्मी से मिलती है, पापा से मिलता है, यानी बहुत सारी चीजें आपकी। कभी कभी आप हूबहू मम्मी जैसे दिखेंगे या हूबहू पापा जैसे दिखेंगे, है ना। तो ये सारी चीजें जेनेटिक है, ये हम मम्मी पापा से लेकर के आते हैं। तो ये चीजें होती है बाहर से दिखने वाली, जिसे हम ओपन आईज से देख सकते हैं, ठीक है। लेकिन जो हम अंदर बॉडी के सेल्स के साथ लेकर आ रहे हैं, जो कि मम्मी पापा के थ्रू लेकर आ रहे हैं, उन्हें हम अपनी ओपन आईज से नहीं देख सकते, है ना। तो बट वो इफेक्ट करती है हमारे शरीर को। तो सिकल सेल डिसीज उसी तरह का एक डिसीज है जो मम्मी पापा से बॉडी के अंदर ब्लड में आया, वो आपको आगे हमारे साइंटिफिक डॉक्टर्स बताएंगे। बट वो मैं सिंपल लैंग्वेज में बता दूं कि मम्मी पापा से आने वाली ये बीमारी जो अंदरूनी है और जिसकी तकलीफ इतनी खतरनाक होती है कि दर्द, हमेशा दर्द के क्राइसिस आते हैं, जिसे पेन क्राइसिस कहते हैं। बॉडी में ये जो सिकल के सेल है वो अगर हमारे जॉइंट्स में फंस जाते हैं तो हमें इमीडिएट पेन स्टार्ट हो जाता है। वो इतना तकलीफ होती है उसकी कि हमें बच्चों को जिसको हो रहा है उसे हॉस्पिटलाइज्ड ही करना पड़ता है। तो जैसा कि मैडम ने कहा है आपकी जर्नी कहाँ से शुरू हुई, तो मेरी सिकल सेल की जर्नी मेरे बच्चे के पैदा होने के बाद से ही शुरू हो गई। क्योंकि वो तड़पता था, परेशान होता था, फिर हम हॉस्पिटलाइज करते थे, बाद में डायग्नोस हुआ 2.5 साल के बाद, और देन पता चला कि इसे ये डिसीज है। तब तक हमें मालूम नहीं था कि ये डिसीज कोई एग्जिस्ट होता है क्या। हमारे आने के बाद पता चला कि ये कोई डिसीज है, और उसके बाद धीरे 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 हम हॉस्पिटल से घर, घर से हॉस्पिटल, हमारी जिंदगी इस तरह से ही चलती रही। और पिछले 4-5 सालों से इस एरिया में हम लोग अवेयरनेस के लिए निकले हैं, ये सोच करके कि ये डिसीज और किसी बच्चे को ना हो, जो कि बहुत आसान है। तो वो किस तरह से ना हो वो बताने के लिए हम लोग निकल पड़े। और अब हम एक संस्था के मार्फत, जो कि नेशनल अलायंस ऑफ सिकल सेल ऑर्गेनाइजेशन जिसे कहते हैं, जो राष्ट्र की सबसे बड़ी संस्था है, जो हर स्टेट में सिकल सेल पर काम करने वाली सोसाइटीज को मिला करके एक अलायंस बनाया गया है, जिसका नाम है नेशनल अलायंस ऑफ सिकल सेल ऑर्गेनाइजेशन। उसके थ्रू हम लोग काम कर रहे हैं और मैं उसमें मेंबर सेक्रेटरी हूँ। तो ये मेरी एक जर्नी है सिकल सेल डिसीज के साथ।
थैंक यू सर फॉर टेलिंग अस। आप सर ये बताना चाहेंगे कि मेडिकेशन वाइज चैलेंज क्या फेस करते हैं, मतलब इंडियन सिकल सेल पेशेंट्स को क्या दिक्कत आती है मेडिकेशन के मामले में?
हाँ जी, सबसे पहले तो इस सिकल सेल डिसीज का परमानेंट इलाज नहीं है। जो आप लोग अभी इनोवेशन कर रहे हैं, जो टेक्नोलॉजी आप लाने वाले हैं इस फील्ड में, जो साइंटिस्ट अभी तय करने वाले हैं, तो वही चीज एक परमानेंट होगी। बोन मैरो ट्रांसप्लांटेशन ही एक फिलहाल परमानेंट इलाज दिखता है, लेकिन ये हर सिकल सेल पेशेंट के लिए आसान नहीं है। इसके अलावा मेडिकेशन हम कहे तो मेडिकेशन ये सिर्फ उसके सीवियरिटी को मेंटेन करने के लिए है कि वो कम से कम हो। तो अगर मेडिसिन पेशेंट को रेगुलरली उसके राइट प्रोटोकॉल में मिलने लगे तो वो पेशेंट अच्छा एक नॉर्मल लाइफ जी सकता है। बट जैसा कि आपने कहा है सिकल सेल डिसीज ट्राइबल एरिया में ज्यादा होता है, हाँ बिल्कुल ट्राइबल में ज्यादा है, शेड्यूल कास्ट में ज्यादा है, ऐसी कम्युनिटी में है जो थोड़े पुअर है, गरीब है, इनके पास साधन संसाधन नहीं होते। तो इन लोगों तक मेडिसिन भी नहीं पहुंच पाती। इन लोगों तक मेडिसिन कैसे लेना है इस बात की भी जानकारी नहीं पहुंच पाती, जिस वजह से जो सिंपल प्रोटोकॉल है मेडिसिन लेने का वो भी उन्हें नहीं मिलता। इसका दुष्प्रभाव यह होता है कि वो बार बार पेन क्राइसिस में जाते हैं। बार बार पेन क्राइसिस होने की वजह से उनके बॉडी ऑर्गन डैमेज हो जाते हैं, और बॉडी ऑर्गन ज्यादा डैमेज हो जाने से उनकी मौत भी हो जाती है। तो हमारे पास मेडिकेशन का बहुत बड़ा चैलेंज इस देश में है। चूंकि भारत देश बहुत बड़ा है, 30-31 स्टेट्स है, उसमें से 14 से 15 बड़े स्टेट्स में सिकल सेल डिसीज है। 15 लाख के ऊपर सिकल सेल पेशेंट है, बहुत सारे गांव खेड़ों में है, रिमोट एरिया में है, वहां तक हमारा मेडिकल सिस्टम नहीं पहुंच पाया है ठीक से। हाइड्रोक्सी यूरिया जो कैप्सूल है जो इनकी लाइफ को अच्छे से रख सकती है, उसकी जानकारी प्रोफेशनल लोगों को ही नहीं पहुंच पाई है, तो पेशेंट्स तो अभी काफी दूर है। कुल मिला के हम लोग जो पेशेंट्स है वो वेट कर रहा है कि कब उनके पास राइट मेडिसिन राइट प्रोटोकॉल के साथ मिले और वो लोग एक अच्छा जीवन जिए। अभी हाल फिलहाल में भारत ने अच्छे कदम उठाने शुरू किए सरकार ने, तो जिसमें कुछ सिटीज या फिर अर्बन एरियाज में ये चीजें उपलब्ध हो रही है, बट रिमोट एरिया और गांव में अभी भी इस चीज की बहुत कमतरता है। थैंक यू मैम।
Thank you so much, sir. Like you said, there needs to be some standardization of protocols and also more awareness regarding the disease, not just at the patient level, but also at the level of clinicians, at the level of clinic centers, Anganwadis, so that they can assist more towards, patients' lives, and they can help those people deal with such a difficult condition. So from this, I would also like to highlight that since actually I'm a recent joinee at the lab, so there's a very small story that I have that I would like to share with the students. So we went on this trip to Raipur, and as you can see here, there are few pictures where some people had come to get tested and at an Anganwadi in Raipur. So during this field trip, I actually got to meet a lot of people in my lab, और जो भी लैब के अभी आगे प्रेजेंटर्स आएंगे, रिधम, मेघाली, and many others I met actually during the trip because it was just a week that I had joined. So मुझे research, translational research जिसे कहते हैं, which is basically from bench to bedside, where whatever you are developing in the lab, whatever research that you are doing on a clinical topic actually reaches the patients in a hospital or a clinic, is what translational research is all about. And I didn't have much exposure of how much impactful such a research could be, and how diverse this field is, and the beauty of science overall, कितना impactful एक topic हो सकता है in changing people's lives, एक disease पर काम करना, एक treatment suggest करना, even किसी भी disease की, just studying the pathophysiology of a disease itself is a big deal if you are trying to help patients out and contribute to their lives in the long term. So I could see that there were many people who were smiling, but they were not really they were just trying to pretend that they were okay with it. But since it was a tribal community, there were there was a very high prevalence rate of sickle cell disease in that area. And as we move forward, I would like to say that initially when researchers join a lab, and scientists, what we call ourselves scientists, when they're working in the lab, some of them just like science because it's just there's a perfection associated with science कि आपको एक हर चीज को बहुत ही step by step you have to do everything perfectly, otherwise the whole experiment goes into waste. So even if you're working for a year on a project, you're working for a few months, or you might be working for 10 or hundreds of, like a long period of time on a research topic, it might be completely a waste of time if you do not exactly know why something is happening, how something is happening, and what could be done to improve a whole scientific situation and discover something new. So there's fascination associated with it, but there are researchers who understand how working on a clinical topic makes an impact onto lives of patients, not just in India, obviously here, but globally as well. So that's what I just wanted to share. Ki mera perspective changed a lot after I came back from the trip. I realized that there's so much beauty to science and there's so much impact we can make by genuinely dedicating ourselves to lab and trying to discover something new in a clinical area is meaningful. It brings an impact to people and we should know about it. So I would like to discuss about the prevalence. As I told already, and as sir explained as well, it's a genetic disorder. It's actually, SCD is an autosomal recessive disorder, meaning that you need to have both mutated alleles, both mutated copies of a gene in order to actually have the condition. Otherwise, it would just be called a sickle cell trait. So in India, the prevalence is quite high. We actually see that there's a 2 to 40% prevalence rate, as has been reported based on various newborn screening programs that are being conducted across the country. Gujarat, mein, Maharashtra, mein, there are many states that are conducting such programs to check ki kitna prevalence rate hai. And also in 2021, I think there were 20 million people alive who had the condition in our country. It's also highly reported in states of United States, in US, and in countries in Africa, in Middle East. So it's quite a prevalent condition. It's the most common disorder, and it obviously doesn't have any permanent treatment. That is, there's no permanent treatment for this in the country currently. And we are trying to develop a treatment that could permanently cure patients of the condition, so that unko bar bar dawai na leni pade, they don't have to go to the clinics again and again. We just work towards a gene therapy that directly targets that mutation in our genetic material, in the genetic material of patients, and it completely eliminates that mutation and thus the symptoms that are associated with it. So let's talk about, so the picture shows actually it's associated with the newborn screening, where you actually see if a child could have a condition or not, and they get a card sort of thing where they're reported as positive or negative for conditions. And let's move to the inheritance. So both parents, as Gautam sir told already, that both parents need to have at least one mutated allele, so as to transfer the mutation, so as to transfer the condition to the next generation. As you can see in the first figure, if both adults have the normal adult hemoglobin is basically what carries oxygen from our heart to various organs of the body. But if that hemoglobin is binding to oxygen, if those RBCs are normal biconcave, then it works fine. But if they turn into sickle shape, that leads to less kind of absorption, less absorption of oxygen, less carrying of oxygen to different regions of the body. So that could result in sickle cell symptoms. And here we see that if a father or if any of the two parents has a normal adult hemoglobin, while one of them has one copy that carries the mutation, then there's a 50% chance that the child would be normal, would not have the sickled mutation, while there's a 50% chance that the child would have a sickle cell trait. That means he's a carrier. He's not having the condition, but he could transfer that mutation to the next generation. And in the next row, as you see that if there is if one of the parents has the sickle cell disease and the other is normal, it doesn't have the disease, so there's a 100% chance that the child would be a carrier of the condition, as in he could transfer it to the next generation. And if both the parents have the sickle cell disease, obviously the child would have it, 100% chances. And if both the parents are carriers, then there's a 25% chance that the child will have the sickle cell disease. So sickle cell disease has different types depending upon the mutation in the beta globin chain, and that results in a differently shaped RBC. So the commonest one is the sickle cell anemia that our lab is working on currently. I've tried to put GIFs so that students pay attention. So I would love it if you guys try to find this interesting, because research is interesting if you are curious enough. So as you can see here, there is if many of you would be able to understand, but for others I would simplify. There's a GAG codon written here, so it actually codes for any kind of a sequence in the DNA, would code for ultimately make up an amino acid. So here GAG actually codes for glutamic acid. But if a mutation is happening, even if it's a single change in a nucleotide that leads to GTG, instead of an adenine, there is a thymine base that is present in a patient who has the sickle cell disease, and that is resulting in the formation of valine instead of glutamic acid. That single change in amino acid results in the sickling of RBCs, and the red blood cells that are healthy would simply move through the arteries and would never obstruct any sort of blood flow. But in deoxygenated conditions where there's low oxygen, or in any such sort of situation where somebody has sickle cell disease and majority of his red blood cells have this sickle sort of shape, then that would obstruct the blood flow to the various arteries. Those sickle cell shaped RBCs would obstruct the blood flow, and that is ultimately resulting in most of the symptoms associated with the condition. So talking about the pathophysiology, there are many acute and chronic complications that could arise because of the condition, because of the mutation. So we have meningitis stroke. There could be acute chest syndrome that is very common in STD patients in India. There could also be gallstones that develop. Priapism is a common disease that is seen, a common symptom that is seen. We also have septicemia, for which you need to be given blood transfusion and need to be immediately sent to the hospital. And Many of these symptoms are chronic complications actually, which stay for a longer period of time. For example, neurocognitive dysfunction, retinopathies, there's pulmonary hypertension. There's also chronic pain. And we also have some people also experience delayed puberty or erectile dysfunction. So based on these diverse symptoms, the treatments that actually exist are very symptom based. So whatever symptom would appear in a patient, the doctors would try to help out directly based on the symptom that comes up. So for example, if somebody has low iron, that person would be given folic acid supplements. Or if there's a requirement for blood transfusion because blood is not flowing because of the obstruction of the RBCs in the arteries, if blood is not flowing to many organs of the body, blood transfusion would be emergently required. And then there's obviously hormonal medicines that could be given for triggering puberty in like children where puberty is delayed. And there are pain medications, as we discussed earlier, like hydroxyurea, 10 to 15 milligram would be advised by doctor to be taken almost every day in order to avoid such symptoms or prevent such symptoms from appearing. But then there is stem cell and bone marrow transplant that also exist in the country, where you will basically isolate stem cells from a donor. As you can see in the figure here, there's a healthy donor who doesn't have any sort of sickle cell trait or sickle cell disease. You collect the stem cells from them, from their blood, and then you kind of process them in a way where you purify and you only take up whatever is required, the RBCs that are required, and you remove the rest of the WBCs and all the other material that is present with it in the blood. Then you cryopreserve it, basically preserve it at very, very low temperatures. You expose the patient to chemotherapy, high dose of chemotherapy and radiation therapy. And basically you are kind of infusing the healthy stem cells into the individual that could compensate for the lack of that fetal hemoglobin, which actually is important to make the RBCs biconcave, to retain the shape of the RBCs as biconcave. So that is the current treatment that exists, but you need to have an allogenic donor for this. Allogenic would be that a brother or a sister, a blood relative, could be a very good candidate as a donor if they do not have the condition. Because if you take up stem cells from a foreign, from anybody who's not in your, who doesn't match exactly, or not exactly, but like so much with your genetic material, then your body might consider the markers on that cell as foreign. So they will reject it. Basically, your immune system would consider it as something that is foreign. They will not accept it if it's not from your brother or your sister or from a donor, a healthy donor that is in your relatives. So it's important to have somebody in the donor that is, so that is a challenge. Obviously, you might not have somebody who's a healthy donor to give. So we need a treatment that is permanent. We need a treatment where we can completely eliminate the condition, and we also need a treatment that can be accepted by the patient's body. They do not consider it as a foreign something that has been introduced and that needs to be destroyed. So it's important that some sort of a gene therapy that could actually target the mutation and completely eradicate the mutation at the root level is important, so that the symptoms don't even arise in the first place, so that the sickling doesn't even happen. So I would like to pass on to Meghali here. She is a PhD student. She's in the final phase of her PhD right now, and she'll talk about CRISPR technology and how CRISPR-based gene therapy is different from the existing treatments that we have for SCD. So Meghali, are you there with us?
Hi, am I audible?
Yes, sir.
Yeah, sure. So hi everyone. I hope you all are doing great. So I am a PhD student working with Dr. Devojyoti Chakrabarti, and I'm for organizing this short seminar sort of for everyone, especially the students, because as we know, you all are the future of this country, and of course we would like you to know more about the different kind of research that is happening in our labs here in India. And coming to the main agenda of this seminar, as Kanika had well explained how the prevalence of sickle cell anemia is so high in our country, and what are the different kind of the ways how this treatment proceeded all over in the world. In the last few years, we have come across this CRISPR technology, which is a kind of more most abundant genome engineering or gene editing tool that has been used in the current years. So what is done in the CRISPR technology is, so let me just tell you the full form of CRISPR. This is clustered regularly interspecies short palindromic sequences. It is a kind of adaptive immune system that is that we have adapted from bacterial system and now can be used for any organisms as such. So what happens is that when, as Kanika had told in her previous slide, that if the the cells do not belong to your uh, immediate relative or family member, then there is a chance of your body to get rejected. It might reject those cells and the therapy might not happen. So in terms of bringing a therapy perspective, what different scientists all over the world have been working on it, and they have seen that when patients, if the cells from the patients directly could be taken, and if a genetic engineering can be done in those cells and that could be transfused back into the patients, then it's the best kind of therapy that could be available to all of us. So CRISPR has made it easy for us. So in what way is that the patient cells, that like the sickle cell anemia patient could they could donate their blood, or the blood could be brought out from their body, and the cells that have the defected hemoglobin gene, those can be corrected with the help of CRISPR technology, with the help of this is a basic representation of how the whole treatment can be done. So the peripheral blood is basically the blood cells that are uh, uh, which are taken out of your body. Those undergo some kind of changes by the help of CRISPR, and those corrected cells could be transfused back to the patients, and that is a kind of a therapy which the patient can undergo for a certain amount of time. And with time, it has been seen in certain places in US, such as in University of California, San Francisco. People have already done these trials in 2 or 3 patients so far, and in the last 2-3 years it has been done, and people with patients basically who had undergone this trial or the treatment, they have seen that they are leading a very normal life than how they used to live before their therapy. So what they do is that in the CRISPR technology, there is a Cas9 protein and a guide RNA, which basically detects those specific region in the genome where it goes and targets that particular region where it is defected, and corrects the T nucleotide to A, and the corrected base then basically makes the normal hemoglobin, and then that can carry oxygen properly. So this is a very useful tool that now a lot of people has been doing, and we also plan to carry it out in our country as a trial, and we want to see if that can help in the tribal population in India for the change, for the kind of change we are wanting to see in future. Besides this, can you change the slide here?
Yes, sure. Um, so, yes, would you like to tell us about the diagnosis, like how it's diagnosed in the country?
It's not changing.
It did at my end. I can see. Can everybody see the change in the slide?
Yes, yes, it is visible.
So, Meghali, would you tell us about the diagnosis, how the disease is
It
diagnosed?
is visible now. Yeah, definitely. So what happens is that till now has been multiple diagnosis processes, like you can do the sequencing of those genes. So basically what happens in sequencing is your gene instrument where it can read the number or the letters of your DNA. So once we do the sequencing, when there is a change from T to A or A to T, that can be detected through the sequencing method. Besides that, there is a very crude method of diagnosis of the sickle cell disease. What happens is that you collect the blood samples, and there is this test called solubility test. So in the solubility test, there is this chemical called sodium diothionate. So what this chemical does is that it chemical does is that it basically, so if your red blood cells are normal, so they will easily break down and you will have a very clear kind of solution. So the tube in the left side, you can see it forms a very light colored solution. Once the sodium thiocyanate breaks down the RBC, you will have trait condition. You will have 50% of normal cells. It cannot break down your
Can everybody else also
it
experience?
cannot break the color of your solution side, you can see. So you can see a clear
We can't hear you
visibility
properly. The voice is
difference in the left.
There's some network issue. She was talking about the turbidity of the solution. From what I heard, that if the solution is that
In the left tube and the right cells of disease, or yeah, am I audible now? There might be some kind of
yeah.
Net
There is some issue and she's trying to tell that there's an issue. She's not able to
normal.
tell. Yeah, there is some issue with the network. Could you repeat the turbidity part? Maybe that's what has not been heard.
Yeah. Is it?
It's better now.
Fine now. There might be some network issue. Okay. Is it?
It's breaking a lot.
Fine now.
It's breaking a lot.
I think probably Rhythm can take over if she has a better network. Rhythm, are you there?
Yeah, hi, I'm here.
Hi. So, yeah, you can take from this side then.
Yeah, sure. Am I audible clearly?
Yes. So should we move to the Rhythm? Move to the
some serious network issue here.
Yeah. So should we move to the CRISPR part first, maybe, and then we can go with the diagnosis? Okay. So then maybe the network improves. So we'll talk about the diagnosis in a few minutes. So let's talk about CRISPR-based Feluda.
Okay. So you can hold to the last slide. I'll start from that only.
Okay.
Okay. Hi everyone. So I am Rhythm, a final year PhD student. So I hope you all have understood some little parts. What is sickle cell anemia? What is its prevalence in India, throughout overall in world, and the symptoms, how it's so severe. The pain is so severe. The patient suffers a lot, I guess. I hope you have felt that. Now, most importantly, we are moving towards therapies. So first of all, it's very important or crucial to diagnose what is sickle cell anemia, or to diagnose the normal human being, to diagnose between the normal and the patient one. So there are already well-established diagnostic forums for sickle cell anemia. So one, Meghali has already discussed about the solubility assay. So first of all, there is a prick that happens in your fingers. So they take out the blood, and in that blood, they add the blood into the solution that has the sodium thiocyanate. And this sodium thiocyanate, if the person is normal, if it does not have the sickle cell blood, that means if there is a lysis of the blood cells and the blood is normal, that has the normal hemoglobin, that has the glutamic acid in it, and then the solution is almost clear. You can see that there is no turbidity. But if the sickle cell lysed and the blood is having the sickle cell mutation, so now the hemoglobin is hydrophobic in nature, and it loses its solubility. That is why it is known as solubility assay. Whenever there is less solubility, that means the solution is turbid, then you can say the person or the suspected one, the person is suspected one. Either it is the sickle cell, completely two alleles are sickle, that means it is patient, and another is the another possibility is that it is having the heterozygous in nature. That means one allele is normal and the another is sickled in nature. So now, to confirm that, there are other techniques. I'm not going to detail or the technical glitches of all, technicalities of all these things, but you can simply understand that it is approximately 2 days whole protocol. Whenever you take the blood and then you do the solubility assay, or at that spot where there is a collection going on, so they take the samples. So they say that you are suspected one. So that person can be heterozygous or it is normal. That can be heterozygous or it is the patient one. So now they undergo second procedure that is known as this chip sort of assay, when they run these samples into the gel, a sort of gel, and whenever they see two bands, that means it is heterozygous in nature, and whenever there is single band on that gel, the person is almost the person is sickled in nature. So now it takes 2 days to inform, or at least 3 to 4 days to inform them that it is approximately you, the patient, you are the patient or you are the heterozygous person. So can you change the slide, Kanika? Kanika?
So we talked about sequencing. Do you want to share about? Okay.
Okay. So, okay, there is one more important, you can say, the strategy to diagnose the sickle cell mutation. As we are talking about mutation, what is mutation? I guess there are 4th to 5th class kids also. So, so you have your body, you have the cells in your body, and cells have the DNA. So everybody has, like, suna hoga DNA kya hota hai? DNA has a stretch of bases. That means there are 4 bases, A, T, G, C. Whenever this A is converted to some another base, you say this is a mutation. So sequencing, as it is suggested from the name only, sequencing means you sequence this whole stretch of DNA. That means, if we are starting from first base, what is the first base? Like it is A, C, G, T, whatever, and then you sequence the second base, third base, fourth base. Aise, this is known as sequencing. So as Kanika has already mentioned about the mutation of the sickle cell anemia, that is GAG to GTG. That means A is converted to T. Then from sequencing only, you can tell to the family or to the person that this has this mutation, GAG is converted to GTG. Then you can say this person has the sickle mutation. But it again takes a lot of money and a lot of time. So what we have done in our lab, we have tried to make a kit, or you can say strips, from this applying CRISPR assay into that to diagnose sickle cell as a point of care diagnostic forum. Why I'm saying point of care diagnostic forum? Because for the point of care, there are 3 things that are important. The assay should be should be visibly to naked eye, like you can see that and you can tell whether this has the normal phenotype or this is the patient one. And secondly, it does not take so much of your time, effort. And the third is it is cost effective and rapid. So there are 3 things: that it is fast or rapid, it is correctly diagnosable, that means the specificity and the sensitivity of the assay should be higher, and it can be used as a point of care anywhere in this whole setup. Like you need very little or less instruments to work to get the results out of it. So I'll be having interactive session. Just a second of interaction I need from all of you. So I'm showing you something. So is it visible?
Yes, yes.
Okay. So in the chat, I need an answer. How many bands in one or two you all can see? There are two strips, I guess you all can see, which is named as 1 and 2. Can you write the number of bands? Just quick answer I need. Someone can unmute also. Unmute करके भी लिख सकते हो. Has anybody answered? I can't see.
Yeah, they're sending direct messages to me. There are many people who have answered. Some say 2, some say 1 and 1.
There are 2 strips. There are 2
Very
strips.
good.
There are 4.
There are 3.
There are 4. There are 4.
There are 4.
There are 4. There are 4.
Ma'am, 4
हमें पता चल गया तुम लोग सो नहीं रहे हो. हमें पता चल गया.
Rhythm, you have also got muted.
Okay, am I audible?
Yes. They muted everybody.
I was checking कि सब active है. हमें पता चल गया, बहुत शोर आ गया, सब सोए नहीं है अभी. So, as you all can see, there are 2 strips, strip 1 and strip 2. और यहाँ पे सबको ऊपर बैंड दिख रहा होगा ना, दोनों में बैंड दिख रहा है, बट यहाँ पे सिर्फ एक बैंड नीचे दिख रहा है, यहाँ पे नहीं दिख रहा. So this means this is positive sample and this is negative sample. So this thing we have tried to develop. So सबको पता है, सबके घर में, सबके not सबके घर में, but जैसे sugar strips होती है, pregnancy strips होती है, वो भी ऐसे ही डिजाइन होती है कि whenever there are 2 bands on the strip, you can say the person is the female is pregnant, and if there is single band that is known as control band, you say this is a false signal. So similarly, we have tried to develop the same thing, but obviously the different chemistry. And if there is no band in this line, we say this is the negative person. And if there is a band niche below me with the control band, we say this is the positive person. So these two, so the strip has a chemistry which I'm not going to show in details, but it uses the CRISPR technology. So we have a DNA. So we take the samples, the saliva samples. We don't take the blood samples. We take the saliva samples, and then we extract the genomic DNA. We extract the DNA from the saliva, and then we do the amplification of that target gene. Like so we have a hemoglobin gene that has this mutation. So we amplify this hemoglobin gene, and then we put the CRISPR-Cas9 complex into the solution. So now in the solution, you have the DNA and you are adding the CRISPR-Cas9 complex. Now with this complex, you put that strip into the tube, which has this complex. So if the DNA is of positive person, that means the DNA has a mutation or the sickle cell DNA, then you see 2 bands, as you can see here. So this is the control and the test band. If the DNA is of negative person, or the person is normal, completely normal, so you see this is only single band, that is only the control band. So why is it named as Feluda? Because Feluda is a Bengali detective. So जैसे ये detective है, detective क्या करते हैं? He or she tries to capture the, जो भी सबने Sherlock सब देखी होगी detective movies. So वो try करते हैं कि वो capture करे. So we have also tried कि हमारा sickle cell और normal completely अच्छे से capture हो without any giving false signals. So the sensitivity and the specificity of the assay is more than 95%. So this is the, you can say, the add-on or the cherry on the cake for the rest of the technologies that has already been developed or already been present for detecting sickle cell anemia. So एक और important चीज है, यहाँ पे हम blood नहीं लेते. We don't extract the blood. We take the saliva sample because blood में So जब हम रायपुर गए थे, I'll share one experience with you all. So जब हम रायपुर गए थे, there are very small kids, like 1-2 साल के बच्चे आप देख रहे हो यहाँ पे. This is me who is trying to take the saliva. So ये बच्चों को बहुत डर लग रहा था. जो ऊपर के तुम्हें दिख रहा होगा, यहाँ पे सब they were all very scared to give the blood, क्योंकि अगर एक आगे वाली लाइन में रोने लगता है तो पीछे सब रोने लग जाते हैं. So it was so hard to take the blood when generally these people were pricking into the fingers. So but you can see this small kid, he was actually enjoying giving the saliva. तो उन्हें मजा आ रहा था कि कैसे saliva spit करते हैं. एक बच्चे ने तो मेरे मुंह पे थूक दिया था, serious. So I said कि just roll on your finger, your tongue, and then spit. He literally spit on my face. So that was a very not nice experience.
Because they ate candy and all before, so it was all.
So yeah, so you can say this is the very easiest way to collect the sample, क्योंकि यहाँ पे you don't मतलब give the pain to the child first at the first stage. So we take out the saliva and then take out the genomic DNA and put the CRISPR-Cas9 complex into it and then add the strip. So if you see 2 bands, that means the person is positive, and if you see single band, that means the person is completely normal. Now, as I've already talked about the point of care, so it is we have tried to make it point of care because kahin pe bhi we can do this assay. We just need 2 machines. एक PCR machine चाहिए होती है and all these complex. उस machine में हम डाल देते हैं and then we add the sample. So this is the थोड़ा सा ये ज़्यादा scientific है, but simply we take the salivary samples and then after collecting the samples, we extracted the DNA or the RNA. So most importantly, sickle cell के बाद, so first 2 years we were trying to diagnose sickle cell by CRISPR-Cas9. So we named it as Feluda. So जब sickle cell का हो गया, then पेंडेमिक चल रहा था पूरा. So हम घर नहीं जा सकते थे, हम टीवी नहीं देख सकते, कुछ नहीं कर सकते थे. तो हमने सोचा चलो एक बार कुछ काम करके देखते हैं. So we were all in labs. So we were trying कि ट्राई करके देखते हैं कि हम कोविड सैंपल्स को डिटेक्ट कर सकते हैं. So what we have done, we have taken the RNA from the patient COVID patient samples. उस RNA को we have again tried to amplify. जैसे हमने सिकल सेल के लिए किया था, similarly we have taken the RNA of the virus and then we have amplified the virus. उसके बाद amplification के बाद we have again put the CRISPR-Cas9 complex. So similarly, if the patient is COVID positive, we have seen, we saw 2 bands in the strip, and if the patient is normally without any COVID symptoms, or sorry, without any COVID virus, we only saw a single band. So then we have applied this to diagnose, I guess, 5,000 to 7,000 samples in a single week, and then we have got the sensitivity and specificity way more than 90% for this assay. And we have also tried to make us this technology friendly and make a app. जैसे स्मार्टफोन में हम नॉर्मली स्क्रीनिंग करते हैं ना, वैसे ही एक तोप से ऐप बनाई थी. यहाँ पे my colleagues made this app. So what you can do is you can just show your, like, जैसे ये स्ट्रिप है, इस स्ट्रिप को डायरेक्टली फोन से स्कैन करो. So you have this app, just open the app and then scan these strips. So then it gives the result, direct result. It is a COVID positive or it is a COVID negative sample. So we have tried this also. So this is the whole journey, you can say, from sickle cell diagnosis to till we got the DCGI approval for the for detecting COVID samples with this strip. I guess this is majorly done. I have majorly talked about the diagnosis. So this is the case of the strips which we are trying to show. Now here you take out the strips and then you add these strips into the solution. So जैसे solution में डालोगे, so in 2 minutes you see the results. So this is a basic chemistry. As you dip the strips into the solution, due to the capillary action, the solution gets moving towards upper side, and you can see the bands are starting have been started developing. So now can anybody tell me कि left or right side कहाँ पे 2 bands दिख रहा है किसी को? On मत करना बस please chat में लिख देना.
Chats में.
Right side या left side?
Left side people are saying, some people are saying right also.
So left side में, on the left side you can see there are 2 bands, clear 2 bands, and on the right side you see a single band. So this we have done it, tried to make a robust time point of care diagnostic for sickle cell, and then we have moved to detect COVID-19 samples. So I'd like to thank all of you to join this webinar and giving us the opportunity to speak about our work or what we have tried to develop or done in the last 2 years. So we'd be happy to take some questions or something, any question regarding the journey.
Any sort of curiosity anybody has, general, specific to the field, love to take up questions.
किसी को research में interest है, scientist बनना है?
There are a lot of hands raising. So
hands raised में hi बोल रहे हैं.
Like to ask something.
Thank you for choosing me. Ma'am, I wanted to ask, like, ma'am, how if the capillary tube is there, meaning the capillary action is there, then it will move upwards, right, ma'am? So how is it coming? Like 2 bands, it will come like that, and then one band for positive, for negative, and 2 bands for positive.
Very good question, Ananya.
I'm Anya.
Sorry, Anya. Okay. So as you have correctly mentioned about the capillary action, so I'll show you something. Is this visible to all? I don't know if I'm visible.
No, actually. You want to share? I should stop sharing then?
yeah, you should,
No.
Kanika, you should stop sharing your slide maybe.
Okay, so I'm visible, no?
Yes.
Okay. So as she has asked a question, very important question, why there are 2 bands in the positive and there is single band in the negative, and why this band is coming lower as the capillary is obviously moving towards upper side. So Anya, so you can see यहाँ पे whenever there is a positive band or the band that is below the control line. So when we add the strip into the solution, the strip has this DNA and the Cas9. This whole complex will start moving towards upper side. So यहाँ पे, so this site has something that holds this whole complex, this CRISPR-Cas9 complex with the sickle DNA. So if the DNA is normal, if the person is pretty normal, it does not have the sickle mutation, then this complex will move towards the upper side. Then where you can see the control band. But if it has this sickle DNA, then this complex will get caught at lower side. Then it's this whole complex start aggregating at this particular site where you see the test band. So only whenever you have the sickle mutation, this is so specific, the lower side is so specific that it catches this whole complex towards the lower side. That is why we named it as Feluda, because it tries to capture this sickle DNA very specifically, and it just let go or freeze the normal DNA. That is why when you have the sickle mutation, it freezes niche below the control line, and you see 2 bands. And if it is a control normal sample, this whole complex will not stuck, or you can say the strip will not attach at the test site. Have you understood something?
Yes, ma'am.
Very
So
nice
you named it, ma'am, did you name the it after the detective Feluda?
Sorry.
Yes,
Ma'am,
yes.
did you?
It is named after the Bengali detective Feluda.
Yes, yes, yes.
Ma'am, he is my favorite character.
Oh, okay.
It is my favorite character too.
Okay, we will love to take more questions. If there is anybody else, one more question maybe.
I want to be an astronomer.
Yashika, you want
Somebody.
to ask something?
You could type probably.
Hello, am I audible?
Yes,
Yes, completely audible.
Thank you, ma'am, for choosing me. I only want to ask you a question that only newborn children get this disease, not adults.
Rhythm, could you hear? Anybody? Meghali, would you like to take up
Yeah. Sure. Dixit is a genetic disorder, as Kanika had said earlier. The genetics of disease, since it is a genetic disorder, you will know that once a baby is born, you can immediately detect it in its genome. So by doing a sequencing or by the solubility test, immediately you can check. So because the symptoms of this disease is it's to know a person is sickle celled or has a mutant, both the alleles have mutated or single mutation is there by since its birth itself.
Okay, ma'am.
Am I
Thank
clear?
you. Yes, ma'am. Thank you, ma'am.
Okay.
We have somebody has asked, Arvind Daksh, I have seen, ma'am, can sickle cell carriers donate blood? So that is quite a, can anybody hear me? Yeah. So where is
Who has asked this question?
It's Arvind Daksh Singh Shekhawat.
Arvind, can you please unmute yourself or?
He's asking.
Yes, Arvindaksh, you can speak.
Yes, ma'am. Ma'am, this was my question that, ma'am, can sickle cell carriers donate blood?
Can sickle cell carriers donate blood?
Yes, ma'am.
Okay, so first of all, thank you for asking this question because it's a very nice question. And preferably not, because if you want to donate blood, then obviously the blood should be healthy. So the recipient or the patient who is taking your blood, you don't know in which situation the person is, because sometimes we just store or give the blood to the blood banks. First of all, whenever you give your blood or you donate blood, there are some criteria to follow. So first criteria is obviously your HBB or the hemoglobin is way more than 9 or 10. And the second is, do you have any genetic disorder? So this is the question in the whatever you say, the performer they have. So ultimately, wahan pe hi you will be rejected to donate the blood. It's not recommended to give or donate blood if you have the sickle trait, because ultimately the blood that is being received by the recipient, it flows into it. It ultimately gets flushed off. थोड़ी देर के बाद तो वो खत्म हो जाएगा, बट उस टाइम पे उसे क्या प्रॉब्लम्स आएंगे, he must be or he or she must be in hospitalized condition or whatever. So healthy blood dena is better, because healthy blood will take the oxygen properly and do all the functions in the proper condition. So better hai, recommended hai, do not donate blood if you have the carrier or you have this sickle cell trait. अगर sickle cell patient हो तो definitely नहीं, क्योंकि तुम्हारे पास तुम्हारा नहीं, जो भी पेशेंट होगा उसके पास इतनी कैपेबिलिटी नहीं होती कि वो ब्लड डोनेट करे, बिकॉज इट्स अ ब्लड, ऑब्वियसली द ब्लड डिसऑर्डर, राइट? सो बेटर है कि you should not donate blood. Very nice question. Thank you for asking.
Sorry. There is another question somebody has asked. It's from G. He has asked Navya, I think Navya Man. So she's asked if from GAG to GTG, glutamine power 6, basically she was asking why 6 is there, why 6 is written. Why, when we say that glutamic, instead of glutamic acid, valine is now being formed because of the mutation. So that's simply the position of that amino acid in the whole sequence that has been referred to. At the 6th position, that amino acid is present. Instead of glutamic acid, valine is being formed. And that's what it meant. When Navya, can you hear me? Is Navya there? Navya Man.
Yes, ma'am.
Yes, so you had asked about the power 6 written with the glutamic acid and the valine. It's just the position of the amino acid in the whole chain. So that is
Okay, ma'am. Thank you, ma'am.
No problem. There is also a question by Lubhanshi who says, why is sickle cell disease prevalent in tribal population? It is by Lubhanshi. Lubhanshi, are you there?
Yes, ma'am.
Okay, so would anyone like to take up? Meghali, would you like to say? Even Gautam sir could talk about.
He's the best person to answer, right?
Yeah.
Yeah, thank you so much. Actually, not only in tribal, but also there is sickle cell disease more prevalence in scheduled caste, other backward classes, Brahmins, Kshatriya, Vaishya. It means our society in few years back, they have screened lot of people, and we conclude that 80 caste having sickle cell disease. So there is no common that only tribal person having sickle cell disease. Now in the India, 1.4 million people having sickle cell disease, and those are from various community. So yeah, there is important that the major part are coming from the tribal area, but not only tribal. Here, all community members having sickle cell disease. I think.
I would also like to add up on in very good question, by the way, Lubhanshi. So in terms of a biologist, I think I would also like to add up after Mr. Gautam. So is that so as we know evolution, right? Evolution has happened since many years, and we have seen since a long years back, mostly the African population, and since where the entire evolutionarily different population have spread out mostly from Africa. So it's like an advantage over your protection towards malaria. So generally what happens is that sickle cell anemic patients have defective hemoglobin. As a result, they cannot have malaria, because malaria again is a parasite that affects your red blood cells, right? So what happens is that whenever there is an advantageous selection in your evolution, there is always a disadvantage along with it, right? So evolutionary certain kind of group or people where had this advantage where they could not get malaria by having a defective red blood cells, but at the same time those patients had anemia, so which at the same time they spread out all throughout different regions of the world. And in India it is more prevalent mostly in the northern belt of India. Specifically, it is also very it's the another reason is also because of malnutrition in the tribal population, or the scheduled caste and tribal part of the India, that is mostly in a malnutrition condition, and they do not really have the awareness of how the entire planning of genetic counseling, and if a person has trait, and if his or her partner also has a trait, and then they end up having a baby that has sickle cell disease. So that is also because of lack of awareness and education, and malnutrition also that plays a big role.
There's also like intra-caste marriages, and that kind of adds up to the whole
Yes.
situation. The many others have asked the same question actually. The abundance in African countries, as you mentioned, and why in tribal population. There's another question that you'd like to take up. It's from Mohit Mishra. He asks, what does it mean if someone has sickle cell trait? Mohit, are you there? I've got many direct messages. I'm trying to get as many questions as I can. Mohit, are you there? Most other questions I can share of abundance in African and Indian tribals.
Yes, ma'am.
Yeah, so you were asking about the sickle cell trait. Sickle cell trait basically means that that individual is a carrier of the condition. He doesn't have the mutated allele, both the copies of the mutated allele. Maybe one of his parents had had a normal adult hemoglobin, while one of the other parent had the other parent was also a carrier. So
Kanika,
as I
you could
explained,
show that slide where you have shown the entire genetics. That would be easier to explain and understand. You can go. So that could.
Yeah, so as we talked about it, yeah, so as you can see here, if you're saying about the sickle cell trait basically means the AS here, which represents a carrier where one copy of that allele is normal and one is mutated. One is mutated from glutamic acid to valine. That change has happened, and that is making that person a carrier. That means they can transfer it. Their children can actually inherit the condition from them. So in this case, for example, as we discussed, the normal adult hemoglobin in one of the parents, AA, while one of the parents has sickle cell disease, has both the copies mutated. So the children have a 100% chance that they would have the sickle cell trait. That means all of them, when they'll get one copy from the father, one copy from the mother, definitely the mother would always be contributing the mutated copy, because she has both the copies is mutated. One of them will go to the next generation. So one of the copies would always be mutated in the child. So child would 100% be a carrier or has a sickle cell trait. While if you see somebody where one of the parents is a carrier, for example, in the lowest column, you can see here AS, which means that the one of the parents has the sickle cell trait while the other parent has the disease, then it makes a 50% chance that the child would have the sickle cell trait. That means he's just a carrier. He doesn't have the symptoms, but he can pass on that condition to the next generation, while there's a 50% chance that the child will develop the sickle cell disease. So I hope I hope your question is answered. Mohit? It's a good question. I mean, sickle cell trait is actually resulting in the high prevalence. Many of the populations in India have the sickle cell trait. About 22 to 40% are reporting the sickle cell trait in Indian population. So it's quite a lot.
मैं इसमें एक बात कहना चाहूंगा. Sorry.
No problem. Go ahead.
कि सचमुच में अगर जब भी हमारे पास यहाँ 500 स्टूडेंट्स देख रहे हैं, अगर हम उनका स्क्रीनिंग करें तो ऑब्वियसली उसमें से 2 to 40% ट्रेट निकलेंगे ही, क्योंकि हमने हमेशा कैंप किया है और हर कैंप में 10% 20% ट्रेट्स निकले ही है. तो ये बहुत ही कॉमन है, आम है. और जितने भी स्टूडेंट्स आज यहाँ पर है, मैं तो उनको भावी साइंटिस्ट के रूप में देख रहा हूँ, क्योंकि वो इस सब्जेक्ट को बहुत सीरियसली लेकर के सुन भी रहे हैं, क्वेश्चंस भी कर रहे हैं. और आप लोगों की मैं सिकल सेल पेशेंट्स का रिप्रेजेंटेटिव हूँ. मैं 14 लाख सिकल सेल पेशेंट बोल रहा हूँ. मैं 2 करोड़ सिकल सेल ट्रेट बोल रहा हूँ आपके साथ मेरे रूप में. तो ये जो मैं कह रहा हूँ वो आपके लिए है कि आप इसमें जो रिसर्च कर रहे हैं ऑलरेडी क्रिस्पर पर, और जो अभी करने वाले हैं, इनमें से कुछ बनेंगे, तो आप लोगों को हम लोग बहुत आशाओं से देख रहे हैं कि कब आप वो चीज लेकर हमारे पास आए, वो जादू की छड़ी जो आप तैयार कर रहे हैं वो लेकर के आए, और हमारे बच्चे और काफी लोग जो सिकल सेल के दर्द से पीड़ित है वो उससे मुक्त हो जाए। दे आर वेटिंग फॉर यू पीपल. You all are intellectual, you are going to be big scientists, big professionals, and we all are poor sickle cell patients are looking towards you. तो हमारी आशाएं आप लोगों के लिए बहुत है, क्योंकि आप कुछ अलग है, आप इस जगह पर है, इसका मतलब आपको एक प्रकृति ने इस बीमारी से पीड़ित लोगों को बचाने के लिए आपको इस थीम से जोड़ा है, ऐसा मैं समझता हूँ। तो इसमें जो भी आप कर रहे हैं वो बहुत अच्छा करें। क्योंकि मेरा अभी नेक्स्ट वेबिनार वापस से शुरू हो रहा है, तो मैं बस 2 मिनट बोलकर विदा चाहूंगा कि आप लोगों का बहुत बहुत धन्यवाद, मुझे यहाँ बुलाया, आमंत्रित किया। यहाँ मैं सिंगल नहीं हूँ, मैं सारे पेशेंट्स के रिप्रेजेंटेटिव होने की वजह से उन सब की तरफ से आपको धन्यवाद देता हूँ कि आपने ये सब्जेक्ट को, जो कि हमेशा से छूट गया था, सिकल सेल डिसीज हमेशा से छूटा हुआ सब्जेक्ट था, जिसको आप लोगों ने आज हाईलाइट किया, बच्चों के सामने आपने इसको बहुत अच्छे तरह से रखा, प्रस्तुत किया, क्योंकि यही जनरेशन आगे जो ब्लास्ट होने वाला है सिकल सेल डिसीज का, उसको कंट्रोल करेंगी, उसको रोकेगी और भारत को सिकल सेल मुक्त करेगी। तो मैं आप सब लोगों का बहुत बहुत धन्यवाद देता हूँ और शुभकामनाएं देता हूँ कि इस काम में आप लोगों को पूरा यश मिले, और हम सभी करोड़ों पेशेंट्स की आप लोगों के साथ बहुत बहुत दुआएं हैं। मुझे आमंत्रित करने के लिए बहुत बहुत धन्यवाद। थैंक
यू सो मच गौतम सर। आपका यहाँ आना हम सबके लिए बहुत ही सौभाग्य की बात थी, और प्लस हमारे बच्चे आपको ये सुन पाए और आपसे एक रियल लाइफ सिनेरियो से जुड़ पाए और उसको देख पाए। आज हमने इस प्लेटफॉर्म के थ्रू इन सभी 500 बच्चों को आपके थ्रू आपसे मिलवा के हमें एज अ कैंप बहुत ज़्यादा खुशी हो रही है, बिकॉज आई कैन सी देयर आर लॉट ऑफ स्टूडेंट्स हु आर एक्चुअली एस्पायरिंग टू नो व्हाट काइंड ऑफ डिजीज इट इज, what are the symptoms, how can they be treated. I'm getting lots and lots of questions on my chat box, and I'm really sorry my audience that I'm not able to cater to all the questions, but my promise to you is that I'll keep forward these questions to the IGIB team and Gautam sir, and I'll request you to be gracious enough to answer those questions so that I can put forward
definitely
to the students. So thank you so much.
थैंक यू। मेरा आखिरी रिक्वेस्ट बच्चों से रहेगा कि आप लाइफ में एक बार अपना जीनोटाइप टेस्ट अवश्य करा ले। प्लीज डू योर टेस्ट, आपके जीन्स में जेनेटिक कोई डिसीज है या नहीं, जरूर करा ले, ताकि ये 500 लोग अगर बचते हैं तो आगे बहुत अच्छा फ्यूचर हम लोग देखेंगे। थैंक यू सो मच फॉर इनवाइटिंग मी। मैं माफी चाहूंगा और जाना चाहूंगा नेक्स्ट वेबिनार के लिए।
थैंक यू सो मच सर।
थैंक यू सर, मिलते हैं फिर। थैंक यू सो मच डॉक्टर रिद्धिमा, डॉक्टर कनिका जी, और आप सभी का, मेघाली जी का थैंक यू सो मच, और थैंक यू स्टूडेंट्स सुनने के लिए। थैंक यू
so much.
Should we take 1 or 2 more questions, Riddhima?
Actually we are I'm so sorry, but we are actually ahead of the time, but we can take one or two questions. I actually have been receiving one constant question from Kanishka Sharma, and the question is that there are two questions. Does gene therapy during embryonic stage cure it permanently? And second is that can sickle cell anemia be detected during the embryonic stage? So 2 questions that I've been getting constantly on my chat box, so I thought I should put it forward.
I think by embryonic stage, she means when the mother is pregnant and you do the newborn, like you test. Genetic. So yes, that could be diagnosed. The mutation could be diagnosed if you sequence the sample, and we could check it in the newborn, in the fetus itself, if they have the mutation. So many people abroad even differ from the pregnancy, and they bring up, they were not mentally prepared for such an answer from a doctor or a counselor, genetic counselor, that their child would have that condition. And since there's 100% certainty, if the mutation is there in both copies, automatically the child would have the condition. So, it's good if people get tested during pregnancy, if you have the opportunity to get tested for any such diseases, genetic abnormalities, and yes, that would come up in a sequencing data. Is there another question from I think we have missed or something?
No, that
There
is
is very one, I'd like to add, there is very one important point that I guess we have missed. So someone, I guess Ayush, has asked a question that can this disease spread through airborne or something like that? So I'd like to add, this is not a communicable disease. So don't be afraid of this disease, because this is a genetic disorder, as we have all talked it about. So it cannot spread through air, water. It's not airborne, waterborne, any commune form. So it's just a genetic form. So whenever your parents or you have this disease, only you can pass it on. So it's a genetic one.
Also,
Here is also one question somebody has asked. Just let me see the name. So, okay, Gargi Ocha has asked, ma'am, if sickle cell trait is not an illness, then why are people tested? Very, very, very important and very like seriously an important question to ask, why people should get tested. It is the same reason why. It's because a genetic disease, it's inheritable disease. So if any of your parents have this trait, like one of the alleles is affected and his or her partner is also affected, then it's highly likely chances that the children, 50% chances of their children to get the disease. So it's very important for them to get tested. All of us should in fact get tested, so that it does not get inherited by the offspring later on.
There's another question from Ayush Saharan. That's the last one I think we should take. He's asked about who discovered the CRISPR technology, who was the founder. So that is Emmanuel Charpenter and Jennifer Doudna. They discovered it many years ago, but they've recently, I think in 2020, they have won the Nobel Prize for that. They're American biochemists and they were working in this area. So
there are 200 more scientists that are working in this field. So these two have not just, but they got the Nobel. But there are
2 people got Nobel, but there are more than 100 scientists who had been working contemporarily at that same time with CRISPR.
A fast-paced field. Everything we get to see, that we talk about things in the lab, we are talking about a research topic, and the next week it gets published. Just because of funding, resources, there are many obstructions at different levels, we could say. But the field is so fast-paced that discoveries happen every week, and that is why for research many people get interested in the field of genetics, and especially medical genetics, not just because it's impactful, but also because the discoveries happen every day, and there's a lot of curiosity at the scientist level to find answers to things and even create therapies for many genetic conditions that are not curable right now. Sickle cell disease is just one of them.
It's almost 5:30 and I'm really sorry, and I'm really feeling very bad that I have to stop this here, and we'll have to move on to the next section. And I'll take all your questions. You can send us the questions on info or camgsc@cam.res.in. I would request my CAM team to post the email ID in the chat box so that everybody can send their questions to this email ID, and CAM will share I'll collate all these questions and I'll forward these questions to the IGIB team, and we'll send back and share all those questions in your student login so that everybody can see and understand what were the questions and frequently asked questions by all our audience here, and they can know the answers which were given by the IGIB team. moving forward, I would like to, on behalf of CAMP, I would really like to thank the IGIB team here, Dr. Kanika, Dr. Rhythm, and Dr. Meghali. Dr. Purti is also here with us, and Ms. and our guest, Gautam ji, who were really, really informative in our session today. I could see and I could analyze, even I was not that much aware of about this disease. I was not aware what are the effects and what are the side effects and what are we doing in IGIB to support, you know, in testing of this disease and bringing the cure for this disease. So I'm feeling really whelmed and overwhelmed to have you on this platform, and so that I could connect and we could connect all of our students here, 500 students here, and they could get such good guidance and awareness about this major illness and major disease that we have been missing out about its awareness in you know, it's a global thing. So I thank you so much for being here on this platform. Thank you so much.
I've also shared a link. Sorry, I've also
Yeah.
shared a link to you guys. It's for the website that our lab has, and you can check about the recent research that we are doing. You can also check about other people's work, other scientists' work at CSR IGIB, and follow on this link and ask us as many questions as you want later with the kind of opportunity you get.
Absolutely, absolutely. So thank you so much. Campaign, please forward that link to all the students. After this session, you can place that link in the student login so that they can visit and they can see and they can read the researches done by IGIB lab. Also, students, if you or any of your friends have missed this workshop or session, please do not worry. The recording of this workshop will be made available under your student login so that you can be, you can revisit and you can see all the slides. Even some of them were asking me if the slides of these of this PPT can be made available. So yes, they can be made available, and second, yes, you can revisit the video and you can see the slides and for your revision. And before we go ahead towards the vote of thanks, few informations from my side. Tomorrow is the third episode for Achievers Leaders of Tomorrow, and it will be made live at 4 p.m. We are inviting our special guest, Master Avi Sharma. So don't miss that episode. If you want to know the journey of Avi Sharma, please join us tomorrow. The link is available in the chat box. I would request my team to please share the link so that everybody can join us. Also, we would love to hear your feedback so that we can, of course, keep improving our content. So please fill this quick workshop feedback form available on the chat box for your reference. Also get the access to interesting podcasts, journals, and articles of CSIR, NISPER, and other CSIR laboratories under your student login. So you can simply visit our CAMP website at www.camp.res.in and go to the student panel, student login, where you can find all the interesting journals under this knowledge sharing session. So moving ahead towards the fortnightly quick challenge, it is the time to announce and post the link for the quick challenge. So one lucky winner will be selected today and will receive a special appreciation from our side. You just have to go to that link and answer one simple question and post some details about you and submit the form. The winner will be announced at 8 o'clock today, so please be tuned. The entries shall be accepted till 7 p.m. So we have approximately 1.5 hours to submit our questions, answers in the form, and the winner will be posted. The name of the winner will only be posted on our social media platform. So do not forget to like, share, and subscribe all of our social media channels. We are available on Facebook, Instagram, YouTube, LinkedIn, Twitter. The links of all the social media handles are you can go and you can like, you can share, so that we can stay updated with all the latest announcements by CAP. Now, this is a goodbye from my side, and I now invite Vishakha. Over to you, Vishakha.
Good evening, everyone. I, Vishakha, coordinator camp, on behalf of camp team, is very thankful to all the students, parents, teachers for being such a wonderful audience. I also thank Miss Kanika, Miss Rhythm, Miss Meghali, and Mr. Gautam, sir, for taking out time to be a part of this wonderful session. Now we will end the session and we'll meet soon with another exciting session of camp. Thank you so much, everyone.
Thank you. Thank you.
Thank you, everyone.